Okay, everyone, welcome to this Friday's seminar. And before I introduce our speaker, I'll just mention some of the events coming up over the next few weeks. This is a weekly event every Friday at 4:15 p.m. If you're not on our mailing lists and you want to be, go to the Oxford University Biodiversity Network events page, and you'll see how to subscribe to that mailing list. But just to highlight some of the things coming up on the Friday sessions, next week we have Earl Ellis talking about the deep global history and transformative future of nature-sustaining landscapes. The week after we have Dustin Benton from the Green Alliance about can we have it all from the land in the UK context? Can we reconcile food and biodiversity and other uses of land in that context? And then we have Christina Hicks from Lancaster. We have Sophie Montserrat from Rewilding Europe, and many other speakers. But additionally, next week we have a additional event next Tuesday, which is with an indigenous group from the Amazon that's coming to talk about their experience of connection to nature during the COVID pandemic and from the Brazilian Amazon. So that's at 5 o'clock next Tuesday in the same lecture theatre. You need to register. It's getting already a lot of registration, so if you want to go, do register for that event and come along to that, and there'll be some sort of a ceremony as well as part of that event. Okay, so on to today's speaker. So Rob Ewers is Professor of Ecology at Imperial College. He's a New Zealander, so he's very stressed today if you follow the rugby at all. He did his PhD in New Zealand and then did postdocs at the Smithsonian and at Cambridge, and has since been at Imperial College London. And in particular, and a particular relevance to some of the work he's presenting today, he was the founding figure behind the SAFE project, which has been this pioneering, multi-trophic, hugely complex and ambitious study in the landscapes of Sabah in Southeast Asia, trying to get a very rich and detailed understanding of an ecosystem as it was fragmented from intact rainforest to oil palm plantation and fragments of forest. I've been part of this collaboration with Rob and his team, and it's yielded a huge amount of insight and data over the last decade or so. So I'm sure we'll hear a bit about that in this context. So thanks for joining us. Over to you, Rob.
Thanks, Vinda. Thanks, everyone. So tropical forests really are quite complicated things, but I'm going to describe all of that complexity to you in 20 seconds or less, which I think I can do. Soil and climate determines the trees. The trees are eaten by herbivores. The herbivores are eaten by predators. Everyone dies. Some of them get scavenged. Others decompose. They become dirt. That feeds the trees. The trees grow leaves. That makes shade, and the microclimate then influences absolutely everything in that system. And that really matters because it means that simple management decisions, like deciding on a logging regime, for example, really touch the entire ecosystem in just 3 or 4 steps. So by restricting the size or the species of tree we log by logging more or less frequently, we can impact the entire structure of this ecosystem. Now, let me take a step back and give you a story of 2 quotes from conversations I had with very different people. So the first was from the director of the Australian Museum, who described to me, he said, natural systems are more complex than the space station and more connected than the internet. And the second is from a senior forest manager in Malaysia who told me that forestry plans in Malaysia don't mention orangutans because that's not relevant to timber management. And hopefully you're seeing the problem here. We have complex systems which have multiple values and in which simple actions can ramify across that entire ecosystem, but they're being managed for individual goals in what I would argue is quite a simplistic manner. So the basic issue here as a scientist is we have no way of helping the forest manager incorporate orangutans into a timber management plan, or beetles, or flying frogs, or pygmy squirrels, or the carbon balance, or the clean water provision, or bushmeat availability. Or we definitely can't help them embed amorphous concepts like resilience and stability into their plans, because those are emergent features of a complex system. And at the moment, we just don't understand that complexity. So the virtual rainforest is our attempt to deal with all of that complexity. And what we're aiming for here is a simulation of an entire ecosystem that can be used to quantitatively guide decision-making. But it's also more than that, because a system-level model like we're aiming for should be helping us understand the system as a system. And we have a broad scientific audience in mind here. So we want it to make sense to animal ecologists and to plant ecologists and to biogeochemists and to hydrologists and soil scientists and climate scientists, basically anyone that you can imagine interacting with a forest environment. We're really trying to create a general ecosystem model. And we have a wide set of user stories that we want to fulfill with this simulation. So we want biogeochemists to be able to use it to quantify the balance and residence times of key nutrients. But we also want systems ecologists to be able to use it to examine and understand emergent features of a rainforest ecosystem, like the origins of forest resilience. Where does it come from and how is it maintained? So we're aiming to construct a one-stop shop for understanding the many and the varied aspects of a rainforest ecosystem. Now, I'll argue until I'm blue in the face that ecology is far too reductionist as a discipline. We boil our analyses down to simple univariate predictors and responses when we know the world is more complicated than that. We settle for analyses that explain a statistically significant 20% of the variance in our data, and we never really question what's going on with the other 80%. We simulate tree communities while forgetting there's a lot of things out there that eat trees. So with the virtual rainforest, we're trying to be holistic rather than reductionist. We're trying to draw together everything that's known about the processes which are connecting plants and animals and climate and soil, and combine that knowledge in a way which hasn't been done before by joining all those hundreds of dots together in a single simulation. And the goal there is to work out how that ecosystem operates in its entirety, for which the use cases are almost unlimited if we can make it work. Now, at the most basic level, this is all about physiology. So the basis of all ecosystem dynamics and the basis of all this complexity is pretty much what you eat, how much you eat, and what you do with what you eat, which in turn is all influenced and feeds back on the microclimate and the soil nutrient environment in which you're embedded. And everything flows from these fundamentals. So that means we're starting with the physiology and the stoichiometry of individual organisms and then joining them together through an interaction network that together forms an arena where those individuals moderate the microclimate, they moderate the nutrient status of the environment in which they're embedded, which then, of course, feeds back again onto those individuals and thereby constructs, recreates all the complexity of the rainforest in one simple step. Yeah, simple. Now, there's two broad categories of things that we're trying to recreate here. So first is the abundances of key plant and animal and microbial functional groups. So exactly how many individuals and how much biomass is there, and how is that distributed across different modes of life, and how do those different modes of life interact with each other? And second, we're attempting to simultaneously close the loops of 5 key cycles. There's energy and water, carbon, nitrogen, and phosphorus. And this is a fine ambition, which I explained calmly and rationally to the ERC, the funding body in Europe, which really prides itself on supporting high-risk, high-reward projects. And I can paraphrase their response with a statement: can't possibly be done. But now with support from the Nomus Foundation, who were willing to take this risk, we're hacking deep into this minefield of taking on this impossible task of taking a tropical rainforest and replicating it inside a computer. So the rest of this talk is going to show you exactly what it is we're trying to replicate. I'll talk you through what we see as the main processes which are driving an ecosystem, how those processes link with those cycles of key nutrients plus energy and water. And then I'll explain how we're trying to replicate those processes in the virtual world and what the challenges are that we're coming up against. I'll build it all around empirical data that we've collected at the SAFE project, which Vinda mentioned. It's a large project I've been leading for the last decade or more, based in Borneo. It's trying to understand the environmental impacts of tropical land use change. It's a loose consortium of hundreds of researchers, each of whom have focused on their own area of expertise. So if you can name it, someone has probably studied it at SAFE. And together it means we've got what could be one of the most comprehensive empirical descriptions of how a rainforest operates. Now, I've tried for this talk, to order all of that information into some sort of structure that will follow through in the talk, starting with dirt at the top and working our way around. But all of the data and examples in this seminar come from this one study site. And it puts us in a really great position to start thinking about ecosystem level dynamics. But before we can do that, we need to provide the arena, the basic arena in which those dynamics occur. So how does the virtual rainforest set up that structure, and how are we going to tackle this problem of picking up that missing 80% of variance? We want it to be good, obviously, and we want it to last. So we're building it using Python, which is fast, but also easy to develop in. It's a language of choice for many of our target end users. We're building it open source, so anyone can dip in and out of any part of the code. And our basic approach has been to use a modular framework that compartmentalizes many of these processes, which will allow users to come in and say, I don't like that part of your simulation, so I'm going to rip that out and plug in my own piece. All of our code has been reviewed by informal panels of disciplinary experts to make sure we're capturing the right processes, as well as getting oversight from professional programmers to make sure that it's built in a way which makes sense and can be robust. And so we're using really quite modern code quality assurance tools through here. So we start by setting up an arena in which the ecology can happen, anywhere from around 100 hectares through to about 40,000 hectares. So that's going from an individual land parcel that someone might own through to a protected area. And we divide this into grid squares. Typically, this would be about 1 hectare. And that in turn defines the size of the plant communities and the size of the resource pools, which is where all the really interesting ecology starts happening. We input a digital elevation model. We use that to predict the direction of water movement through the landscape. And we pull down large-scale climatic data from online sources, and we downscale it to match our arena. So that allows us then to use climate scenarios as forcing data to start exploring future outcomes in the system. And with that virtual arena set up, we then start to plug in the ecology, which all starts by thinking about the first items on our diagram here, which is dirt and how it connects to water and plants. So the physical environment is our starting point. We've got some dirt, we've got some air, we've got some sun, there's a climate. That's going to set limits to how fast or slow the system can work. And it's not a fully blank slate, of course, because there's a whole heap of nutrient context for our system, which is here. It's a bit obvious to point out the dominant source of carbon and nitrogen is going to be the sky. But the soil is a lot more interesting, probably. Contains a whole heap of goodies. And without going into that whole rabbit hole of microbes just yet, we can say the soil has got this important stuff in it. It's got the different forms of nitrogen, which plants can take up. So you've got some nitrates and ammonium. There's the orthophosphates, so the phosphorus sources that are available to plants. And there's also water, of course, which came from the sky, but has a whole lot of that stored there in the dirt. And then we get our first life. The very first seedling arrives, and it's not much, but it's the base of the power system. It's photosynthesizing. It's taking energy from the sun, carbon dioxide from the air, converting it into biomass. And there's an important side effect here, which is that it starts releasing water through evapotranspiration, which straight away connects what the plants are doing with that water cycle. And it's one of the first complexities we hit with something like a virtual rainforest. Somehow trying to simultaneously simulate microclimate hydrology in plants introduces some real difficult timing issues, because plant responses to soil moisture are rapid, but we don't want to be simulating things on the scale of seconds or minutes. So somehow we have to be averaging these responses over longer timescales. And there are rapid dynamic feedbacks to worry about here. So for example, evapotranspiration changes the vapor pressure deficit, so one measure of the amount of water which is in the air. And that in turn has an instant effect on the rate of photosynthesis, because if you have drier air, you get slower photosynthesis because plants start closing their stomata to save water. And it all creates a chicken and egg problem for a simulation like this. You know, does the leaf release water vapor first and the microclimate then changes? Or is the microclimate there first to influence how much water gets released? And in the real world, of course, these things are happening simultaneously. But in the virtual world, we have to make difficult decisions about what happens first and what happens second. And that can influence the outcomes of a simulation. Now, to simulate microclimate, we're using a model that leverages first principles physics, and it predicts temperature, so including the soil temperature profile, relative humidity, atmospheric carbon dioxide, wind profiles above, within, and below the canopy. This approach involves solving differential equations for each canopy and soil node, which compute storage and simultaneous exchanges of heat and vapor between the various layers. And all of that's happening on a sub-daily time step, grid square by grid square. So these particular processes are operating really at local scales within a grid square. At large spatial scales, there's another connection here, though, which is through rainfall. So that rain comes down, some of it's getting intercepted by the leaves, sitting on the leaves before it evaporates away. The rest is getting to the ground, either directly as throughfall or indirectly through things like stem flow. That's replenishing the water supply in the soil, which the plants are already tapping into. But when there's excess water, so more than the trees are using, it's going to end up in your streams and rivers. Now, most of that's going to get there through subsurface flow, but there's also surface flow, which is what drives surface erosion and also is a big, heavy contributor to floods. And what I want to quickly highlight here is a link between the amount of trees we have and how fast that water starts getting into the streams, because that's what is going to determine when floods happen. And this is something we want to be predicting with our simulation. So Anand Naina showed this effect by looking at the time between a rainfall event and the time at which a stream has its peak storm flow. And the more trees you remove from the habitat, the quicker you reach that peak. So the y-axis here is on a log scale. So we're talking about really substantial differences where the time it takes for a flood to happen in a primary forest where you've got lots of trees, it's almost double what it takes when you go to an oil palm plantation where you have not many trees. So this is something we simulate using a grid-based hydrological rainfall runoff routing model. It's all based on the list flood model, which is widely used in operational flood and drought forecasting for policy support. And it gives us a really excellent starting point. Now, that movement of water through the soil towards the rivers physically connects several of the main nutrients that we care about to the water cycle as well, because, of course, some forms of nitrogen and phosphorus are soluble. So if you have a lot of water flowing through the soil, it picks these up and dumps them in the river, a process called leaching. And it's one of the reasons for why we have detectable amounts of nitrogen on the left and phosphorus on the right in the water of even really small headwater forest streams. That doesn't vary very much between a primary and a logged forest, but it is hugely variable within a stream, probably depending at least in part on how recently it rained. But regardless of that within stream variability, we can confidently state that nitrogen and phosphorus are being constantly leached out of these systems. And of course, not all of it's being leached out. Plenty of it's being picked up and used by the living things of the land. There's two dominant groups extracting it. One is the microbial community, so bacteria and fungi. The other, of course, is the plants. And sometimes those two groups are going to work together to extract those nutrients. Mycorrhizae are that obvious example here. Speaking in gross generalizations, though, microbes and plants are using these nutrients, but in different directions. So, for example, the plants are taking up nitrate or ammonium through their roots and turning inorganic nitrogen into organic nitrogen, Well, the microbes, by and large, are doing the opposite. They're taking the organic nitrogen out of the dead stuff, turning it back into the inorganic forms, which the plants can then pick up and use. Which brings us to the next bit, which is about how plants grow. So we start back with our seedling, but this time I'm going to explore more of the ecology rather than the physiology. So start with our seedling begins small, but it has to get big, for which it has to get through a couple of ecological hurdles. So first, the seed needs to germinate, which means it has to avoid being eaten. And we know if you throw a seed out in a forest, there's an 80 to 90% chance that an insect or a rat or a bird or something is going to come along and pick that seed up and have a little nibble. But if your seed does happen to survive that step and it does germinate, then it still has to avoid being eaten while it tries to establish itself. And this can be a real problem for some species of tree. So this is data for the Borneo ironwood, which we suggested in this paper, it might actually be suffering from complete regeneration failure driven by seedling herbivory. In particular, it gets feasted on by the sambar deer. There's a classic density-dependent element to this, and that if there's a lot of you in a patch, you tend to get focused on and browsed more heavily to the point that survival becomes close to impossible. Now, these two data sets combined, one on seed predation, the other on seedling herbivory, illustrate the magnitude of the barrier that an individual plant has to cross to establish itself and win the opportunity to grow. But if it does make it, then it can start growing, which is essentially an accumulation of carbon from the atmosphere, nitrogen and phosphorus from the soil. And those nutrients are being combined into a form of biomass that we look at and we say, there's a tree. Now, we simulate this using the P model developed by Colin Prentice, which balances carbon gain against water loss, and it estimates the optimal behavior to maximize gross primary productivity. Then there's an allocation model that sets out how different plant functional types will allocate that productivity to respiration and growth, which lets us simulate how trees are changing in height and in diameter. And the heights of the tree cohorts within a cell gives us a canopy model using perfect plasticity approximation, from which we can then work out things like light transmission through those canopy layers. Now, there's quite a lot of tree growth happening. So around 13 to 16 tons of carbon are being soaked into every hectare of forest every year. That's our estimate of net primary productivity. It's roughly the same in primary forest as it is in logged forest, although it doesn't all end up in the same part of the tree. So some of it will go into trunk and woody parts, some will go into roots, some will go into leaves and the canopy, for example. And that allocation depends on what type of tree you are. So in logged forest, for example, where you get more fast-growing early successional trees, on average, there's more allocation of carbon into the woody biomass, which is kind of what you'd expect as the trees are trying to grow high fast. Whereas primary forest, you get a higher allocation of carbon into the leaves, which again makes sense because the trees there are by and large quite tall already. If you're going to keep competing, then you just need more leaves to get more light. And this means one of the key tests for the virtual rainforest is whether these large-scale emergent properties of plant communities are going to emerge from the collection of plant functional types and the allocation models that we use. But these are the sorts of patterns we're hoping to replicate. There's another place where plant-obtained carbon ends up, though, which is outside of the plant itself and in the soil where it's promoting microbial activity. So Andrew Nottingham's girdling experiment showed this really well. So he went out and ring-barked a set of trees. And they were trees which were going to be clear-felled anyway, so he was taking advantage of this to get some amazing data. And these figures show how the amount of soil carbon fluxes, so the y-axis on these figures, it declined by about a third. You go from a few months before the girdling on the left here to a few months after the girdling on the right. So it's really astonishing. It's suggesting around 1/3 of soil microbial activity is directly sponsored by nutrients coming from the trees themselves. So it's a really powerful force driving the soil microbial systems. And that's about all I want to say about trees. So let's start working up the trophic levels here. Trees and plants now are solely the base of a giant pool of resource to support consumer biomass, and that starts with the herbivores. Like a plant, a herbivore is born small, it has to get bigger, and how fast they get bigger depends on how much they eat. So it's essentially a transfer of biomass. It's the movement of carbon and nitrogen and phosphorus from the plant into the animal. And a lot depends on how efficiently the animal is able to convert that biomass from being a plant into being an animal. Those consumption rates are driven very heavily by how big you are. So this data is for mammals. It shows how the distribution of energy intake varies among animals of different sizes, from little ones at the top to big ones at the bottom. And the numbers here tell you how many species of mammal are in each of these size categories. And basically, you've got elephants and pigs and things which eat a vast amount. But then there's a heap of rodents and bats and civets and so on that are small but numerous. And because they're small, they have very high metabolic rates, which means they have to eat a lot. And so they're also having a really heavy impact on system-level energetics. And of course, there's a wealth of information about how the metabolic rate of individual organisms scales with body size and also with body temperature. These are global phenomena that are really well described empirically. And it means we have a clear physiological map to figuring out the daily nutritional needs of any given animal. So we're simulating the dynamics of the animal community using an agent-based model with animal cohorts where the fundamental rates are all derived from allometries and metabolic scaling relationships, which in turn allows us to trace the impact of perturbations like temperature changes and see how they might work their way through the food web. Again, as with plants, animals allocate those nutrients to different parts of their body and for different reasons. So some of it's used just to power themselves. Some of it's used because you have to build a skeleton, which takes a lot of phosphorus, for example. If they're doing well and they have a surplus of food, it might get allocated to reproduction because the female has to grow enough biomass that some of it can essentially be extracted from her body and then gifted to her offspring. But herbivores have a very different chemical composition to plants, and that introduces one hell of a problem that we call stoichiometry. So in a nutshell, that means the ratio of carbon to nitrogen to phosphorus in what you're eating doesn't necessarily map on to the ratio of carbon and nitrogen and phosphorus that you need to survive and to grow and reproduce. So animals have to be clever and picky about what they're eating. So depending on their particular physiology, they might have to specialize on certain foods. They might do better eating fruits instead of seeds or leaves instead of wood and so on. And that's because the stoichiometry, that ratio of carbon to nitrogen and phosphorus, the stoichiometry of these different parts of a plant are themselves all different. A plant is not a homogeneous mix of nutrients. And what suits a particular herbivore best is a match between their stoichiometry, the stoichiometry of their food, and all weighted by the food which is available to them where they are. And animals do vary their diet according to what's available and what they need, which Matt Jarvis showed by collecting and sequencing the feces of long-tailed macaques. In a primary forest, they were eating a huge amount of Lepidoptera. Around 2/3 of their invertebrate intake was moths and butterflies. Whereas in the plantation, almost half of their arthropod prey is comprised of ants. So it's an easy example, really, of how a generalist omnivore is switching their prey according to what's available and what they're needing. Now, a herbivore is so vastly different from a plant that they're never going to have a perfect stoichiometric match. So this possum somehow has to turn their snack of bananas into more possum. And to do that, they're going to have to eat far more of one type of nutrient than they want, just to get the amount of the limiting nutrient that they need. And that's pretty much the basis of poo. So animals get rid of the excess nutrients through feces and urine, and they have to do this to maintain a stoichiometric homeostasis. And of course, all of those feces, all of that urine hits the ground where it becomes the base of a brown web, which I'll come to in more detail soon. But before I get there, I want to quickly mention that animals have another trick that they can use to maintain their stoichiometric balance, which is they can get up and they can go somewhere else, because animals move, even sloths move. And one of the reasons they move is so they can find a new location with a food source that's better stocked in whatever nutrient it is that they're lacking at that particular time. So in the virtual rainforest, we're assuming that nutrient limitation is the single key driver of animal movement. One of the interesting complexities that comes out of animals moving is that when they start running around a landscape, they might not be alone. This is the basis for seed dispersal, of course, which is a really important connection between what the animals are doing and what the plants are doing. Now, mostly animals don't move seeds all that far, only a couple of meters from our seed tracking experiments, depending on whether it's a big seed or a small seed or a fleshy seed or a hard seed, and whether you're moved by something small like a rat or something big like a porcupine. But it really is an important couple of meters, because I've already shown you that seedling mortality and seedling herbivory is density dependent. And we've used some simple simulations to show that the few extra meters of dispersal that an animal gives a seed can be enough to get that seed away from its siblings and result in quite large gains in terms of survival probability. So before I dismiss plants to go focus on herbivores, now I'm dismissing herbivores. They're simply food for predators now, which is our next step up the food chain. Predators are just animals, though. So the same basic patterns and processes exist for herbivores, for predators, as do for herbivores. But where predators have an advantage is that the stoichiometry of a predator is much more similar to the stoichiometry of its prey than the stoichiometry of an animal is to a plant. And that makes being a predator a whole lot more efficient at translating prey biomass into predator biomass than a possum will ever be at turning bananas into more possum. But the predators have different problems, though. For example, their prey move and they have to chase it. And that prey capture and handling costs energy and sets a limit to how much prey they're able to consume. And pretty much that's all I want to say about predators, really, because I think if we've cracked the herbivore problem of how they grow and reproduce and so on, then we've also pretty much simultaneously cracked the predator problem. So I want to skip from there, that very basic discussion of trophic levels, so plants and herbivores and predators. I want to skip from there and move on to the next step, which is thinking about a food web, so a bit more complicated connection amongst these animals. Now, animals vary in thousands of different ways. You know, there's body size, there's trophic level, there's habitat strata they inhabit, there's physiology, sociality, so on and on and on. At SAFE, we've got data on about 7,000 different taxa, which, without even really trying, we've categorized into 120 different functional groups. That diversity starts building in a lot more complexity to the virtual rainforest. But ultimately, all we're doing here is narrowing down the food choices and refining our information about metabolic rates, which is all information that we already have and which is already coded into the virtual rainforest through our general processes. So essentially, all this food web complexity is coded in by just looking at who eats who. So, for example, ground hunting ants can only attack ground-dwelling prey, like this poor old cockroach. If you're nocturnal, you can only eat food that you can find at night, and you're only really going to be prey for an animal which is out hunting at night, and so on. A social animal requires enough food to feed a group rather than feeding an individual, and an omnivore, like this Malay civet, can eat a wider variety of prey than a strict frugivore. But in all of this, all we're doing to add food web complexity to the virtual rainforest is two things. So first, adding or removing energetic constraints. So for plants, something like the rate at which they're able to conduct photosynthesis, and particularly when they're in the dark understory of a forest. For animals, it might be through their body size and whether they're an ectotherm or an endotherm. And the second thing we're doing is narrowing down prey and diet choices. So Dave Hemprick-Bennett showed, for example, that some bats consume a wider variety of prey than others. And it's true for many other groups. There's a gradient from specialists through to generalist consumers. And by accounting for this in the virtual rainforest, what we're doing principally is refining the flows of carbon and nitrogen and phosphorus from one functional group onto the next. But it also sets the scene to allow for competition, where you have two functional groups which might exploit the same resource. So here's an example of competition for access to invertebrate prey, which might be accessed by vertebrates like birds or other invertebrates like ants. And we know there's competition here because if you do exclusion experiments, then you get a reduction in the rate at which insect predation occurs. And it tells us that both invertebrate and vertebrate predators are competing with each other for access to that same prey source. So ultimately, what we're aiming to do here is have a simulation which lets us build up a picture of the total energy flows in the system, in the forest, at the level of a system, which we did in this paper empirically, led by Yudvinda. And it showed us that energy transfers, so this is the size of the circles here, they're dominated by insectivorous birds in both primary and logged forests. So birds only have about 1/3 of the biomass that mammals have, but because they have such small bodies and such high metabolisms, they account for more than 2/3 of the total energy flows in both of these systems. And this is another example of the sort of emergent patterns which we're hoping will emerge from the virtual rainforest simulation. So now to start trying to finish off the circular economy, we need to talk about how things die and get turned back into dirt. So decomposers like this chunky little millipede here, they're really key functional group. They're absolutely critical because every single thing which grows must at some point decompose. And it's not just a straight biomass thing, of course, remember, because all of that dead stuff itself has a stoichiometric ratio of carbon and nitrogen and phosphorus, and all those separate nutrients themselves have to decompose and be recycled. There's three main resources that get fed into the pool for decomposers to work on. The first is dead stuff, whether that's plant or animal based. Some of that decomposes really easily. So take this orangutan, for example, it fell out of the canopy, it didn't survive the fall. Some of this is easy to decompose, which we know because here's that exact same orangutan 6 days later, and it's already reduced to a pile of bones. Bones, though, are really quite hard to decompose, and they contain huge concentrations of phosphorus that somehow has to get recycled back into the soil. And that is one of the mysteries which we're not quite sure exactly how that happens as yet. The second resource for decomposers is excrement. So feces are the obvious, but we shouldn't forget urine as well. Poo is famously food for dung beetles. There's plenty of them around. So Claudia Gray showed that at any given location in a logged forest, you should expect to find around 40 species of dung beetle belonging to 4 functional groups. And, you know, if you're out in the forest and you need to, and you do a dump in the forest, as every field researcher has done, then about 40 grams of it is going to disappear every day, thanks to the magic of dung beetles, who like nothing more than cleaning up after you. Okay, the third resource for decomposers is stuff that fell out of the tree, but without the tree dying. So this figure is not total net primary productivity, which I showed earlier. This one is instead is just showing the portion of that productivity that's accumulating in the canopy. So there's a whole heap of leaves and twigs and fruits and seeds that comes raining down out of the canopy and has to be decomposed. Anywhere from about 3 to 6 tons of carbon per hectare per year, which is hitting the forest floor, has to be turned back into soil. And then by using our known ratios of carbon to nitrogen to phosphorus of plant leaves, it's safe, we can estimate that there's also around 200 kilograms of nitrogen and 100 kilograms of phosphorus coming from the canopy and turning over at the same time, just from the canopy alone. which starts bringing us to the unavoidable, which is microbes. They're the group which are really critical for decomposition, and they represent one of our biggest challenges because we just don't know about them in nearly as much detail as the rest of the system. So it's by far, it's the hardest to figure out what the actual processes are, and also to parameterize those processes is a huge challenge. But we do know they're important because we can see what they do. So Sabina Both ran a litter decomposition experiment, for example. It showed that microbes are more important decomposers than invertebrates. So she showed it by first taking litter from a forest and chucking it back out in that same forest where it came from. And then it decomposes, obviously. So we're measuring here leaf mass loss over a 6-month period. So higher values on this figure mean faster decomposition. If you exclude invertebrates at those same sites, then the rate of decomposition doesn't change much at all. It drops a little bit, but only by a small fraction, which suggests the job is being done by something else. And by process of elimination, we're left with the microbes. Where you do get a role for the invertebrates is if you take litter from one place and you drop it in a new location. So take litter from a logged forest and drop it in a primary forest, for example, or the other way around. Here, the rate of decomposition does slow down when invertebrates are excluded. And it's because the microbial community at the new site isn't adapted to this weird type of litter that it's just had dropped on its head. So what we're suggesting here is that microbes really do dominate the leaf decomposition process when those communities have been given an opportunity to adapt to the leaf litter which is present. But if something goes off and changes in your environment, then invertebrates, which might be a bit less specialized to particular forms of litter, they can then start picking up a larger role and keep that process running until the microbes have had a chance to adapt to the new situation. And in a similar experiment, Daffodilias also detected what we call this home field advantage for microbial decomposition. He showed that the most chemically recalcitrant litter, so litter which has got the highest nitrogen, lowest nitrogen, highest lignin, and so on, they all decompose faster when they're combined with home soil and home adapted microbial communities. It's a really powerful force. And of course, we can see just how busy the microbes are by looking at how much gas is coming out of the soils. So that includes nitrous oxide in logged forest soils, like this study by Julia Drewer, or carbon dioxide, like this study by Terry Riota, which also worked out where that carbon dioxide is coming from. So some's from respiration of wood and roots and mycorrhizae. But by far, most of the carbon dioxide coming out of the soil is from microbial action. That's breaking down soil organic matter and pumping it out into the atmosphere. And it matters a whole lot because if you look at the full carbon budget for our forests, let's say primary forests are carbon neutral, but logged forests are carbon sources. They're emitting more carbon into the atmosphere than they're soaking up. And that's driven very strongly by microbial respiration, decomposing leftover organic matter that the logging events left behind, even though the logging may have happened a decade or more ago. So we obviously need to model microbes. We're doing this using an enzyme explicit resource pool model, where the pools represent different forms of key nutrients, so things like protected carbon or microbial biomass carbon. And the exchanges between these pools are modeled using a system of differential equations. The big advantage of going enzyme explicit like this is it allows new functional groups to be added more easily, because microbial functional groups, to a large extent, can be defined by the specific enzyme or set of enzymes that they produce. And we want to keep ourselves with the flexibility to add additional complexity to this part of the simulation in the future. And that's how we're tackling this seemingly basic process of how to decompose a fallen leaf, which is really so fundamental to making solid predictions about the emergent features of a rainforest ecosystem. It also closes the loop, brings us right on back to that pile of dirt, which was our starting point. So that's the system trying to replicate. Now I want to that into a virtual rainforest. So we're focusing heavily on consumer resource interactions, whether that's plant, soil, herbivore, plant, predator, prey, decomposer, dead stuff. They're all a class of the same basic consumer resource interactions. And we have 4 pieces of information that we're building the simulation on. So first, we know what the functional groups are. These are our consumers, whether that's a plant or an animal or a microbe. Second, we know the body size and we know the body temperature of those consumers. Third, we know the stoichiometry of those consumers and, of course, the prey or the resources which they're consuming. And fourth, we know the climate and the basic soil status of the arena in which everything is operating. Now, metabolic theory uses body size and temperature to tell us everything we need to know about the daily energetic costs any given individual organism faces. Stoichiometric theory tells us what we need to know about how effectively the carbon, nitrogen, and phosphorus in a resource is translated into the tissue of the consumer. And well-established links and feedbacks between plants and water and climate tells us what our arena looks like in which these individuals are operating and how they in turn impact that arena. So essentially, as long as we know how big you are, roughly what you eat and what the outside temperature is, and that's all knowledge that we do have, then we can apply these rules to simulate the life cycle of any given individual consumer. And then it's a matter of joining the dots. We know the stoichiometry of each of the consumers on any given day. We know how hot or cold it is. That means we know how much energy and how much carbon and how much nitrogen and how much phosphorus you need to eat on any given day, as well as what's in your body should you get predated and be consumed. We know who eats who, and we know who's passing which nutrients to who and how many of those nutrients we're being passed. So that gives us the basis to simulate the system in its entirety and from the ground up. And that's the approach we're taking to tackle this impossible challenge and break it down just process by process and step by step until we get there. And it's not to say we don't have serious challenges, because we do. This is very much a work in progress. And we really do have some big problems we're facing, which I'm just going to run through 3 of our perennial favorites. Appropriate time and space scales of different processes, and how do you integrate them all into the same simulation? We've got everything from microbes and microclimate operating on the time scale of hours and spatial scale of millimeters, through to generation times of trees measured in decades and centuries and over square kilometers. Beyond that, we have the timing and sequencing challenges I mentioned earlier. So these show up in a number of places, like the timing of consumption versus the timing of production can really strongly influence the outcome of simulation. And somehow we need all of these processes to talk to each other at temporal and spatial scales and in ordered sequences which make sense, which is something which is really easy to say. I just said it to you in 20 seconds, but it's really quite devilishly difficult to do. We're going to run into computational limits as we try and catch ever finer details about functional groups. So we don't know how finely we can divide up plant and functional groups yet. And even more, we don't really know much about microfauna in the soil and how we're going to integrate a soil food web with an above ground macrofauna food web. And then, of course, there's bacteria. What the hell do you do about bacteria? We've no idea how finely we're going to have to resolve microbial functional groups to get ecologically reasonable outputs. We just don't know yet, but we do hope the answer is 2. And finally, what do we do about things which leave the boundary of our simulations? Simple things like frogs, which dump a whole heap of reproductive mass, so eggs. They go and dump them into a stream. But at some point in the future, that has to be returned into our forest as a new cohort of little frogs. So that was 3 headline examples of the types of challenge that we're facing up to, and that we need to deal with if we're going to successfully turn the rainforest into a simulation, which, if it all goes well, we can then start using to better understand and to better predict, and then to better manage our ecosystems. Right, acknowledgements. This is a big, complicated project. It involves a big team. These are the people that are really driving it forward. A lot of them are here today as well. They're all co-authors on the seminar. They've all got their individual parts of the project which they're tackling. And I really want to take some time to recognize their contributions and to encourage you all to talk with them over a glass of wine at the end of this seminar once I shut up. Beyond them, I'd particularly like to mention Glenn and Ryan and Ed and Johnny, who were central to collecting the reams of data which I've presented. And of course, there's a huge number of students and postdocs and collaborators that have been part of SAFE. A whole cast of people beyond the ones on this slide have all contributed to the data I'm presenting today. And of course, what I've been doing has been expensive, but I have had really amazing financial and institutional support, particularly Sime Darby Foundation, who supported SAFE, and more recently, the Nomus Foundation, who are supporting the Virtual Rainforest Project. I'll leave it there. Thank you.
Okay, can I open up to questions?
That was a really interesting talk. Thank you very much. I know you've mentioned that it was maybe one of the wicked problems that you might be running into, but I was wondering how you might be assuming some of the microbial or fungal metabolic rates that you would need to introduce into the model to make it accurate.
I'm going to pass that straight to Jacob.
Speakers on the room. It's struggling to hear everything. I mean,
Oh.
you can't hear these questions on there.
Yeah, but the speakers aren't on, it's not the microphone, it's the problem.
Something not on. We've been like this.
You use this speech both, it's got one's for online, one's for.
Okay.
Well,
Use both. Okay. Okay. So is that the question was how are we assuming the rates of the fungi and
different functional groups.
so I think we would be trying to derive it based on sort of like metabolic theory of ecology approaches. Exactly how you do that will be tricky because you're doing across entire kingdoms. So with changes in temperature and stuff, we'd use that metabolic theory approaches kind of thing. That's it. Oh yeah, we have to average across enzymes. So I think we'll have to do quite a lot of digging in the data to try and find what's a sensible kind of average value for this is an enzyme that decomposes carbon produced by bacteria. What's the kind of average rate constant, thermal response, all of that stuff. And then I think we're just going to do absolutely loads of parameter sensitivity stuff because, yeah, they are major unknowns. And I think, yeah, that's sort of the approach we have to take, I think.
Yeah, no, I'd love to chat more about it.
Yeah. Cheers.
That was so much easier than answering it myself.
Ooh. How's that for volume? Is that all right? Sounds very loud to
That's
me.
Lindsay.
Great talk, Rob.
Oh.
Great talk, Rob. Thanks very much. So how are you going to go about sort of testing how well your model's doing? Yeah, that's it, basically.
Yep. So you want to wait for the mic? But they were nice pictures though, weren't they? So validation, yeah. When we first started mapping out what our system looks like, we ended up with something like 100 or so physical features, if you like, of the system, which can be measured with empirical data. And I haven't lined it up properly, but I reckon we've probably got physical data on more than half of those, which we can use to validate the simulation. And the simulation itself, we're trying to build it on global parameters rather than ones which are taken from our empirical data at SAFE. We're trying to keep all of our empirical data dry for validation to the extent that we really can. And so the goal then is just to have predicted against observed for as many of these different aspects of the system as we can. And one of the key features of that is that if you can imagine coloring your predicted versus observed dots for, this is to do with animals, this is to do with microbes, this is to do with microclimate. One of our key things we're really wary of is that if one of those colored blobs is way out, like we don't want a system which is working well for 3/4 of the processes, but completely haywire on the other one. So I think one of the fundamental measures of success is whether we're getting equal predictability across the entire system.
Okay, so what's been, so I know you're still some way along the development journey. What's been some of the biggest surprises you had in trying to do this holistic simulation?
Do I need that?
Yeah,
Oh, I've got. I think the amount of time that it takes to just set up a model architecture and a model core, like even before you start thinking about the science, just having a framework in which you can plug that science in and have all these different parts of the system talk to each other, and setting up an arena, having a data object which all the different modules can pull data from and put data back into, like those sorts of, it sounds really dull. I don't know. I think most people probably do find it dull. It's taken huge amounts of time just to set that basic computing infrastructure up before we can even really move on to the science. And I think we all underestimated how much time that was going to take.
And the science, as you've been developing, obviously you haven't got the simulations running. Any surprises in your thinking? Kurt?
I don't think there's been real major surprises, but it has thrown up some interesting conversations, particularly these timing and sequencing issues. But also, I mentioned bones. I don't know how many hours we've talked about bones and what the hell happens to a bone. You know, some of these little nuggets which are in the system, which you often just never really think about. It's been really fun to, you know, stop and point at them and say, what the hell happens there, and go digging into those bits.
Hi, I really liked your presentation. Thanks. And congratulations to the whole team. I was really curious and interested because something I'm kind of looking at in a different angle is like the above and below ground stuff, and like how you reconcile that and how you communicate that to other people, like outside of science as well. But I'm also I work in the Amazon rainforest, and like the complexity of this blows my mind, because I kind of think to myself, even within a very small area in the Amazon, you're going to have like tons of different ecosystems. So one of the ecosystems I've been working in the last 3 years is our peatlands, and like honestly it's like a whole different world. And when you talk about something like the bones or decomposition, you know, how fast it will decompose, how long it will last, how it will contribute to the sort of infrastructure and the legacy, as you put, of that system is so difficult as well because of the conditions, when it's raining, when it's not raining. How do you envisage reconciling those complexities of so many different kinds of ecosystems in such a small space, like in a rainforest area, and above and below soil, or dirt, as you call it?
I've been told off for calling it dirt before. Yeah, it's a really good question. So we're trying to build something which is general. We're not trying to build something which only works at our one study site and doesn't work anywhere else. But we are only working on processes which are relevant to our study site. So for example, you mentioned peat soils. What we're doing wouldn't work on a peat soil because it's a whole set of different processes which we don't have built in. But we would hope that what we would have as a structure where, because it's modular, you could then slot in those peat specific processes, or you could go to a temperate system. You could plug in some, you know, freeze tolerance and what happens when you go below zero, like things which we don't care about at this point because it's just not part of the system. So in that sense, the simulation should be generalizable. It's just a matter of checking it's a sense check really, the processes which you expect to be in your system and whether they're in the simulation, and just identifying what you need to plug in to make it work in your new system. In terms of connecting above and below ground, one of our big discussions actually feeds into Yavinder's question. One of our big discussions is when do things stop being a microbe and start being an animal? Because the microbes are being dealt with by Jacob, the animals are being dealt with by Taryn, who's not here, and the approaches to those two groups are really quite different, the fundamental modelling approaches you use. And so we still haven't resolved that giant grey area of when you go from microbes and microbivores up through nematodes and the mites and earthworms and so on, and at what point they become animals which come out of the microbial system and go into the animal system. We don't know. Don't know yet. How hard can it be? Feel like I should walk around with the mic. Maybe I should. Maybe I'll start doing that.
Thanks. I'll be quick because I know you're interested in starting the weekend. But you know, when you reduce entities and you simplify, you know, you lose things. And I'm thinking especially when you go from, you know, a full community to biogeochemical processes and physical factors. And I'm wondering what this modeling effort misses, and especially, you know, the species themselves. And you know, thinking about Aoife's question too, the you know, beta diversity between communities, or you know, just the interactions of the species themselves at, you know, different trophic levels, and you know, how can you sort of account for that and think about that in these models? So what do you end up missing and in these sorts of approaches?
I'm going to walk around with this for you now. So we definitely lose species level resolution. We're not trying to do that. We're trying to do functional groups. So you don't have species level competition within a functional group, for example, is an obvious thing we're missing. What we are aiming for, though, is to be able to investigate specific target species, which you could then code as their own functional group, with just such a narrowly, tightly defined set of parameters that it is just this one species, because we want to be able to put orangutans inside a simulation where the whole forest is working, for example. But I think that's probably the obvious thing we're doing, is one of the places where we're just stopping, is we're not going down to modeling every single species in that system.
Okay, just both.
Okay.
Thanks very much. And sorry if I also came a little late, so I had something to do with this question, but I was just wondering if you could talk a little bit more about what would be maybe your ideal way you'd see this model being used, and what might be some unideal ways it might be used. Because as a non-modeler, you often hear how, well, models, the only thing they have in common is they're always wrong. So it was interesting that, you know, you were talking about calibrating it and trying to get it right, but what are some of the risks? Because, you know, we can never quite be sure that it's right. How this might be used, that's going to be positive, or what might be some of the risks involved?
So in terms of use cases, it really depends who you ask. So we have a set of user stories, 9 or 10 different user stories that we want it to work for. So I mentioned a couple of them early on. So things like a biogeochemist should be able to use it to estimate residence times of key nutrients, for example. A manager should be able to use it to try and get a management plan to optimize whichever part of the system or multiple parts of the system thereafter. Guarding against ignorant use is a whole different kettle of fish. It's not actually something we've talked about directly. Maybe we should start having conversations about that. But it's, in that sense, it's no real different to, did that just turn off? it's no really different to any other type of model. Like the model has some processes in it. It has a limit to detail. If people are using it without stopping to think about whether their outcomes and what they're trying to understand is actually replicated in the simulation, then they're going to get erroneous stuff. I don't know the extent to which a model developer can necessarily guard against that, because we want this to be open source. We want it to be used, but ultimately users themselves have to take a bit of responsibility for themselves. It's an awkward connection point because we want it to be easy to use, but the easier you make it to use, the more likely the situations are that you're describing. I guess the main thing we can do to guard against that is clear documentation. So we're documenting as we go. So at least users have the information they need without having to read the code and understand the code. So they at least then they don't have an argument for ignorance.
So I just wanted to ask how detailed or how complicated your models are for animal movement, and in particular whether they can whether they're sort of detailed enough to capture things like landscapes of fear, for example. Or will they just be sort of the agent finds where there's the most food and stays there?
It's very much food bottom-up driven. So we're not doing things like predator avoidance, certainly not now. Wherever we can, we've tried to shy away from behavioral processes to think more about what's driving demographic processes. Just because then you get into a nightmare, an extra nightmare world. And I think we've already chewed off quite a big nightmare world as it is. So yeah, landscapes of fear and so on. It's not there. I mean, I can see 3 years from now when everything else is working, it would be something which could be plugged into it, but it's certainly not on our immediate radar. And most behavioral things are not in there.
I'm going to just shout. Thank you, Rob. I thought it was absolutely brilliant talk, completely inspiring, and to the whole team, even though I've strained to hear because I was so bloody fascinated by it. Thank you. What advice would you give to any of us who might want to do this for a system? Where do you start? How do you start thinking about the level of complexity that you need? I know that's a huge question, but if you had to give 1 or 2 crucial pieces of advice, what would they be?
I don't know. I would say don't do it alone. And I would say be extremely careful and extremely picky in getting your team together. Because I think when I think of the sorts of people I collaborate with, I collaborate with Yadvinder. It's because Yadvinder knows a whole heap of stuff I don't know. I don't really collaborate with people who have overlapping expertise with myself, because you just get clouded into minutiae. Whereas if you have very different people with different experiences and different knowledge bases, you get much more progress. So I think that would be my key thing, is to get the smallest group of people you can with the expertise you need to cover the breadth of what you're aiming for. Tip number 1.
I'll take that for starters.
So just to say about I find it absolutely fascinating. It reminds me, I work a little bit with system models, and have been since the 90s. Where you try and model the biosphere and the atmosphere and the hydrosphere and bring them all together. I think some of the challenges you're facing in this one are ones that I'm familiar in that context. That in every little section, you can go deeper and deeper. You can try and model clouds, and you can try and model droplets, so you can try and model turbulence, and everywhere there's an infinity of depth you can go to. But actually having the breadth to try and bring it all together at some level, and try and find those trade-offs between complexity and just having a go. You know,
Mhm.
part of the thing is just to have a go, because and defy those ERC reviewers who like, say this can't be done. Because I think that, as you highlight, there are still huge challenges, and there's always this finding this fine finesse between how much complexity is enough for the questions you're trying to answer. But it's really impressive what you and your team are doing. So congratulations.
Thank you.
Right. So we have drinks just down the corridor, so feel free to come and join us for drinks and snacks and a conversation. And let's talk to Rob before 8.
Amazing. I love that.